Hello, my name is Devin Rance. This is my presentation topic. It's the use of gene drives to mediate disease resistance in endangered species. So, the establishment of a globalized society has been actually paralleled in modern times by the development of a globalized ecosystem as well. However, despite the inherent benefits it has had on us as a species, it has been inherently detrimental to species across the world. Now, the reason for that is that these newly formed connections between disparate ecosystems across the world has so often led to the propagation and spread of disease from these pathways back to other ecosystems. These situations have often mediated an increase in the pressure applied to species in different ecosystems, often pushing them towards either extinction or further endangerment. Now, most of these species that are often found in different ecosystems usually lack the ability to even resist against these diseases. That has actually opened up an interest in the scientific community on this topic. The scientific community is currently trying to use gene drive technology and CRISPR to actually insert genes, resistant genes, our genes, into these endangered species to mediate resistance to these diseases, which could possibly restore or save these endangered species from extinction. So that actually brings me to one of my main topics here. So what is a gene drive? So in its simplest definition, a gene drive is a genetic engineering technique used by researchers in conjunction with CRISPR systems to insert a desired gene into a host, as well as insert the CRISPR genes as well. The point being is that these CRISPR genes have actually been designed or programmed to actually insert that desired gene back into the host post-reproduction. So, when you put it into your host, as soon as it undergoes sexual reproduction and has offspring, the offspring will have that gene, and so on, and so on, and so on, until it propagates into the whole population eventually. So, that is just crucial for the topic that I want to talk about today, which we'll get to that. But I think this diagram provides a great kind of example of the comparison between a standard population with no gene drive and then a gene drive population where you can clearly see that it is slow, basically it spreads throughout all the offspring from that original host. So that is just crazy. Now that we've talked about how gene drives work, let's talk about how disease affects endangered species. So diseases, despite what people may think, are not the direct cause of most extinctions. Typically, disease is like a symptom to endangered species. It applies additional pressures on a species which slowly push it towards extinction. It's a contributing factor or variable that leads to it, not the cause. So, why is that the case? So it has a lot to do with how disease affects populations on a local and global level. So at a local level, disease is a main driver of the direct decline of a population. However, at a global level, it's not. It's a very infrequent driver of extinction. So, how does disease actually directly affect a population of an endangered species? Well, it's pretty simple. It directly affects the biodiversity of that endangered species, which is just crucial because if you think about it, if it affects the biodiversity, these populations are already pretty small, so any additional factors or pressures on them will lead them down the road to extinction. So how does it reduce biodiversity? Pretty simple. It's two factors. It reduces reproduction or stops it completely, and it kills the host. If the host can't pass its genes on, then you lose genes in a population. And if it can't reproduce, well, it can't pass those genes on. So two primary examples of this is the American chestnut, which has been reduced by a fungus that is commonly referred to as chestnut blight, and it can no longer reproduce out in the state of nature, and the American elm, which is being affected by the Dutch elm disease, which also kills the host. So that is kind of how disease directly affects endangered species. So what about species in general, in terms of like how disease affects them? So this figure right here shows you species stretching across different types of animals that are being affected by disease. Now all these are endangered, or it's basically each category that we currently list species under, which is least concern, near threatened, vulnerable, endangered, and then critically endangered, and then finally extinct in the wild, and then extinct. So these graphs clearly show that certain species are more threatened by disease than others. Obviously, the more critically endangered something is, it is more liable to be affected by disease, which is pretty clear based on biodiversity. So that's kind of how disease affects endangered species. So that brings us to the main question here. How do gene drives influence mediating disease resistance in endangered species? So why is it a good idea? So it's pretty simple. So CRISPR by itself, inserting a resistant gene into a species, is already pretty revolutionary. But couple that with gene drive technology, where you give the ability to give an endangered species not only a guarantee of survival by giving it a resistant gene, but you guarantee not only that sole host survival, you guarantee the population and future individuals of that population a guarantee at survival. Because the way gene drive technology works, as displayed in this figure here, shows that once it is introduced into a population, it will spread throughout that whole population within 12 to 24 generations, or a few years to a decade. That is crucial and incredibly quick to happen within a population. Now, there are issues with gene drive technology. So say you put a gene in a population and this gene has some negative side effects. Like it may lead to this endangered species, dominating a certain ecosystem. There are methods to actually make sure that doesn't happen. So you can have daisy drives, which basically you have a chain of effects or repairs or genes that actually counteract each other and check each other. So it actually removes a gene over a certain amount of generations, which actually still gets the gene within the population, but it removes it over time, so it doesn't remain in the population, so any negative side effects can be eventually cut out, if that makes sense. So despite that issue, this is just crucial for saving endangered species. It's just effective, and it will do the job that it needs to, and it can do it quickly. And that is crucial for saving endangered species. So that brings us to the most atypical example of how gene drive technology can actually save an endangered species. The American chestnut is a great example of this, because about a century ago, it was the dominant tree that inhabited most of the eastern forests of the United States. It was known to be the largest, the tallest, and the fastest growing tree within these woods. But also, in addition to that, it was a keystone species because it had a very stable, consistent nut season where it produced its nut harvest, which a lot of species relied on that. In addition to that, it is an important heritage tree to the United States. Personally, where I grew up at, in Appalachia, this tree was harvested and cultivated for a large nut harvest to sell around Christmas. In fact, there's several poems and songs that mention American chestnuts, including a Christmas song written in 1946 that opens up with chestnuts roasting on an open fire. I'm sure you guys have all heard that before. But how did it actually disappear and decline from our woodlands around here? It's pretty simple. Turn the 20th century, Americans started importing and introducing the Chinese chestnut here to the United States. In 1908, the fungus that causes chestnut blight, called Trinetria parasitica, had an outbreak in 1908, and every year since then, or since that moment for the next 40 years, it spread throughout the entire range of the American chestnut. And by the time 1950 rolled around, the American chestnut was effectively extinct. It couldn't, or functionally extinct, because it couldn't reproduce in the wild anymore after that point. And the main reason for that is the fungus attacks the adult stage of the tree, which juvenile trees can't reproduce, so basically it functionally could not actually reproduce in the wild. So the American chestnut disappeared from the American wilderness. And except for a few pockets, it was basically gone. However, since 1990 and earlier, for some societies that revolve around the American chestnut, the State College of New York's College of Environmental Science and Forestry, ESF, and the American Chestnut Society teamed up together to actually produce a resistant cultivar of American chestnut, able to withstand American chestnut blight. So they actually proved successful at finding a very effective gene to actually mediate resistance to chestnut blight. The gene was called OXO. It came from a gene found in yeast. It produces a enzyme that breaks down oxalate acid, which is the primary weapon used by chestnut blight to actually attack a tree. So if you can break down that compound, you can therefore effectively fight off the fungus. So this method was successful, but it had a key flaw. It had to rely on outcrossing and several generations of crossing to keep the gene within a population. But the problem was that out of 111 specimens that were tested, only about 56 had the gene after the generations of crossing. That's about a half of the population. That's not effective at providing resistance for a whole population. So that's where gene drive technology can actually come in and fix that issue. So gene drive technology is crucial here because it can fix that problem. So now the methods used to insert CRISPR into the American chestnut is very similar to how they were doing it in the actual experiment I was just talking about. But once you introduce this gene drive into one host and then have it propagate and start producing offspring, this resistant gene stays within the population and it's not outcrossed. So it stays in the population and gene pool, so every offspring gets the gene, which effectively means that the population is resistant. It's resistant to chestnut blight, and that allows researchers and arboriculturists to actually bring this tree back, which is just amazing. So you can actually restore a species by putting this gene in and effectively making a whole population resistant. it is mind-blowing that we can possibly do this, and I think we honestly should. We can introduce this tree species back into the environment.